Hello friends, today I am going to reveal some true facts of mechanism of remembering. But first of all, I request you to please share and subscribe my channel for getting my informative true facts videos uninterrupted. Please note that subscription of my channel is free of cost. Now, scientists decodes mechanism of remembering and forgetting in fruit flies. Researchers have shown for the first time the physiological mechanism by which memory is formed and then subsequently forgotten. The research which was done in fruit flies looked at the synaptic changes that occurs during learning and forgetting. The investigators found that a single dopamine neurons can then drive both the learning and forgetting process. It is a common expression to say that your brain is full, although the brain doesn't literally fill up. In recent years, researchers have discovered that the brain does something push out old memories in order to take up new ones. Brain does something and sometimes push out old memories in order to take up new ones. Now a team at Scripps Research has shown for the first time the physiological mechanism by which memory is formed and then subsequently forgotten. The research which was done in fruit flies looked at the synaptic changes that occurs during learning and forgetting. The investigators found that a single dopamine neuron can drive both the learning and forgetting process. The study was published in Cell Reports. We believe this system is set to remove memories that are unimportant and not necessarily supposed to last a long time, says first author Jacob Berry, a postdoctoral associate in the Department of Neuroscience on Scripps Research Florida campus. He said, I find it elegant that all of this is done with the same neurons. Our paper highlights exactly how this is achieved. To study memory in flight, the insects are conditioned to associate a particular odor with an electric shock. Once they have trained scientists to observe that they subsequently avoid that odor, which confirms that the memory has been made. By monitoring the activity of neurons in the brain before and after the conditioning process, scientists can get an inside look at the physiological underpinnings of memory formation. In earlier work, the Scripps research team showed that there are specific dopaminergic circuits that are involved in both the formation of memory and the removal of memories. In the current study, the investigators and imaging techniques to look at the process in more details. They discovered that when behavioral memory is degraded, the cellular changes made during the learning process are reversed by the same dopamine neurons that helped from the changes in the first place. The researchers also found that when the dopamine neuron is recruited to form a new memory, it also works to degrade older memories. Whenever you learn something new, you are simultaneously forming a new memory, while potentially interfering with or erasing old ones. Berry said it is very important balancing act as it prevents you from becoming overloaded. It prevents you from becoming overloaded. For decades now, neuroscientists studying learning and memory have focused on how the brain acquires information and how that information is made to be a stable memory. A process called memory consolidation, says first author Ron Davis, a professor and chair of the Department of Neuroscience at Scripps Research. Only recently have neuroscientists grasped the importance of active forgetting and begun to unravel the processes that causes the brain to forget. Berry adds that this learning and forgetting process helps to explain the retroactive interference, a common observation in psychology. Retroactive interference describes the situation when more recent information gets in the way of trying to recall older information. For example, calling our former boss by our current boss name. Although the research was done in fruit flies, the investigators expect that the findings will apply to higher organisms, including humans. Evolution worked out a lot of important processes like this pretty early or berry seeds. So there is a lot of relevance to studying these synaptic pathways in simpler organisms. The study led by Berry not only provides new insights into the brain mechanism for active forgetting, but offers a wonderful example of how much we learn about brain functions from laboratory animals like the fruit fly, Drosophila, David seed. Understanding the process of both remembering and forgetting and potentially how to manipulate them has a number of implications for humans for conditions like drug addiction or post-traumatic stress disorder. It may be beneficial to develop approaches that can boost active forgetting, improve memory retention. On the other hand, could help to treat dementia and other forms of memory loss. In addition to Davis and Berry, Scripps research postdoctoral associate Anna Fan contributed to the paper. Dopamine neurons mediate learning and forgetting through bidirectional modulation of memory trace. This study was supported by the National Institute of Health. In my next video, I will reveal some true facts of other very interesting current topics. Thanks.